Welcome to Ajat Bioacademy. Today I will talk about neurotransmitter loading or neurotransmitter packaging, or you can say neurotransmitter storage. As I said in my previous video, that is, and the whole process of neurotransmission, I told at the time I will make separate video for individual part of the neurotransmission process. So today's part is neurotransmitter loading, or you can say neurotransmitter packaging, or you can say neurotransmitter storage. So this is the neurotransmitter loading, you can say this is packaging, or you can say storage. Okay, so as I said earlier in my previous video, that is, neurotransmitter will be loaded into the synaptic vesicle, and that vesicle was basically two types: large dense core vesicle and small clear core vesicle, or you can say small synaptic vesicle. Okay, that means within the vesicle the neurotransmitter will be within the vesicle, that means will be storage. So if I just think this is a neuron, and this is nerve terminal, this is dendrite, and you can say this is soma, and this is axon. So this, in that terminal, the neurotransmitter will be loaded into the synaptic vesicle. So if I just draw in different way, that is, you can say this is the neurotransmitter terminal knob, and here will be the neurotransmitter vesicle, and this is the postsynaptic part, and in that area, presynaptic terminal, the vesicle will be there, and the neurotransmitter will be loaded within the vesicle. So if you think this is the neurotransmitter, this is the neurotransmitter, and that will be loaded here, and after that, after activation of the nerve, it will release the neurotransmitter. That is the neurotransmitter release. But today's topic is loading, how it will load into the vesicle. Okay, so just come to the main point. So today's topic is neurotransmitter loading. So firstly, you have to think a vesicle. So if you think this one is your nerve terminal, and this one is your vesicle, synaptic vesicle. So vesicle is protein in nature, and that will actually come from the soma. It will, the protein in nature substance will be synthesized within the soma, because in soma all the protein manufacturing machineries are there. That can be ribosome, endoplasmic reticulum, Golgi bodies over there. So the vesicle will produce there, and it will travel down into the terminal, and here the neurotransmitter will be loaded. Mind it, in my previous video I've also mentioned there, that is, most of the neurotransmitter will synthesize within the nerve terminal, because here lots of enzymes are there. But protein in nature neurotransmitter will come from the soma. Okay, and also that small neurotransmitter also may synthesize within the soma, but mostly synthesized within the nerve terminal, and after that it will be loaded. Today's topic is how it will be loaded. So what does it mean, the synaptic vesicle? Synaptic vesicle is, this is the protein in nature substance, and this is the synaptic vesicular membrane, and that membrane is embedded by different types of channels, different types of pumps. Other membrane protein are there. Here one thing will be there, that is the, you can think this is a pump, and there this pump is nature is ATPase. That means if I name this name, this pump is VATPase. V for vacuolar, not vascular. Firstly discovered from the vacuole. So vacuolar ATPase, and that VATPase will be embedded within the membrane. And what is the function of VATPase? The VATPase, the main function of VATPase is to make the entry of proton from outside to inside. So if you think this is your proton, okay, and when the pump will be activated, and then what will happen? The proton will enter. Before going to the details, I want to add something more about the VATPase. So VATPase is a pump, not truly a transporter channel. So pump means it's a protein embedded in the membrane, and that is attached with a particular protein which has a enzymatic activity, and that part is ATPase. So from the name you can think what will happen. It will just change the ATP into the ADP, and that will release inorganic phosphate, and it will produce energy. Okay, that means ATPase, that means it will activate the ATP to produce energy, and that energy will just work to open the pump, and that pump will enter proton from outside to inside. You can say this is the proton pump. This name is proton pump. So what is the name of the proton pump? Is VATPase, vacuolar ATPase. Okay, so this pump is embedded within the on the membrane of the vesicle. Okay, another important things are also embedded within the vesicle. That is, we can think a channel, that is a antiporter channel. You can normally say transporter channel. So why it is called antiporter means something will enter and something will exit. So you can think this is neurotransmitter will enter and the proton will exit. Proton will exit through this channel. So this is called, this you can name it neurotransmitter proton antiporter channel, or simply you can say neurotransmitter transporter channel. Okay, transporter simply, or you can say transporter channel. So these two types of substance, protein substance, are embedded on the membrane and very crucial elements of the vesicle to load neurotransmitter into the vesicle. Other substances are there, the snare protein, different types of snare protein for docking and fusion, and also some muscular protein to control or regulate the this function of transporter or pump. Okay, so now this is neurotransmitter vesicle. Already I said it can be large for protein in nature neurotransmitter, and for small it can be small. So commonly it is called synaptic vesicle. Okay, so that vesicle, the loading process is basically two types. Basically two types. What are the types? One is co-packaging, or you can say co-loading, normally, and second one is independent packaging, independently loading. Most of the cases, a single type of neurotransmitter will contain multiple number of vesicle, and that vesicle will carry a single type of transporter, and single type of neurotransmitter will enter into the vesicle. This is very common. Suppose if you think this is the dopaminergic neuron, so dopamine will produce within the nerve terminal, cytoplasm or cytosol, and it will enter from inside to outside through a single type of transporter channel. Okay, but some nerve terminal, some vesicle can retain or can contain more than one type of neurotransmitter. Co-transmission, earlier in my previous video already I said. So this is co-packaging. So co-packaging mostly, already I said, that is a single type of neurotransmitter will go through the channel, a particular channel, because the channel and the neurotransmitter has a affinity, okay, specificity. So over there, through that channel, the neurotransmitter will enter. So if it is just done by the co-packaging, then what will happen? A single transporter channel, through the single transporter channel, different types of neurotransmitter will enter? Mostly no. It has some exception, like GABA and glycine can enter through a particular single transporter, but most of the cases another transporter are there, will be embedded. Suppose this is another transporter, antiporter channel, and through this way another type of neurotransmitter will enter and a proton will exit. Okay, suppose if you think this is the glutamate channel, so the name will be different. I will just roughly I will mention here, that is muscular glut transporter channel. And if you think this is the GABA, so the name would be muscular GABA transporter. Okay, that means a single vesicle can carry two types of neurotransmitter. So this is the co-packaging, and it will basically done by two different transporter channel, some exception also. Okay, so now what will happen? Now this is the co-packaging, or what is the independent packaging? Independent packaging means separate vesicle will be loaded through a separate neurotransmitter channel. Okay, that is the point. Now come to the main mechanism of loading process. So if you already know, so this is the cytosol, or you can say nerve cytoplasm, cytosol, and this is inside, this is outside of the vesicle, and this is inside. So in inside vesicle, the vesicular lumen part. This is the vesicular lumen, or you can say inside. Okay, normally in normal condition, the cytosol pH condition is 7.2, and inside pH is 5.5 to 6. In normal condition, the pH is 5.5 to 6. That means initially inside the vesicle is acidic, and outside of the vesicle, that means cytosol, is slightly basic. Okay, that means in normal condition, inside the vesicle, the number of proton is more than outside. Okay, So then what will happen? This pump, that is VAT phase pump, is initially or during its actually origin, you can say, it is constituted. That means always open. Okay, automatically it will work without any influence. That means you can say when excessive neurotransmitter will be synthesized within the nerve terminal, then excess neurotransmitter will activate them and then it will open. Not like that. It will automatically work. That is constitutive pump. Okay, continuously it will activate and it will work. So what will happen? When this pump, that is VATPase pump, will activate the ATPase activity, it will show the ATPase activity, then it will turn the ATP into ADP and it will increase, it will produce energy. And that energy, by the influence of that energy, through this pump, that is proton pump, the proton will come from outside to inside. Then what will happen? So now excess proton, more proton will be inside. So what will happen? The inside of the synaptic vesicle will be more acidic. Okay. How? By the help of VATPase. So when VATPase will be activated and the ATPase will produce energy from the ATP, that energy will open the pump and it will enter the proton from outside to inside. So inside proton will be more and it will be more acidic. In that case, the acidic condition will be 5 to 5.5. 5 to 5.5. Initially 5.5 to 6, and when more proton will enter, then it will be more acidic. Okay, then what will happen? That means inside the vesicle, more proton. That means chemically, one gradient is already occurred, already, or a gradient already created. That is chemical gradient. Okay, and you know the proton carry the positive charge. So positive charge is more inside than outside. Okay, so another gradient is electrical gradient, charge gradient. So two type of gradient will be there. Will be there. That means electrochemical gradient. Electrochemical gradient. So when electrochemical gradient will develop within the vesicle throughout the membrane, electrochemical gradient, here chloride ion and some in some extent sodium ion can influence the gradient, electrochemical gradient, but main role, the proton will play the main role to create electrochemical gradient. Okay, and that electrochemical gradient will be created by the help of VATPase pump. Okay, so what will happen? By the influence of electrochemical gradient, the antiporter channel, that is neurotransmitter transporter channel, or you can say neurotransmitter proton antiporter channel, will be activated. So that channel is nature is inducible. That means someone will induce it and then it will activate. So which one is the influencing matter or factor? The influencing factor is electrochemical gradient, and that electrochemical gradient is basically comes from the influence of VATPase. Okay, so here when electrochemical gradient will just influence the pump, influence the channel, then what will happen? The neurotransmitter, normally you know the neurotransmitter is present more excess outside of the synaptic vesicle, and inside the vesicle is 0 or very negligible, and gradually, as it is always open, so gradually it will work and neurotransmitter will enter and enter, okay, by the help of electrochemical gradient. Okay, so in that way gradually it will enter. Then what will happen? A time will come, the neurotransmitter will be more inside the vesicle. At that time the neurotransmitter will be less in cytosol. Then what will happen? By the influence of electrochemical gradient, from the low concentration neurotransmitter will go to the high concentration, and that means again is the concentration gradient at that time by the influence of electrochemical gradient. From the beginning, electrochemical gradient is just working there, and gradually it will make the entry of neurotransmitter from outside to inside, and a time will come when inside the vesicle number of neurotransmitter will be higher and outside will be lower. So from lower concentration to higher concentration, the neurotransmitter will very quickly enter by the influence of electrochemical gradient. So electrochemical gradient is directly connected with the ATP? No. Here this VATPase is directly connected with the ATP. The proton pump is directly connected with the ATP energy. Okay, so the energy and the gradient, proton gradient, initially it is conducted, it is just carried out by the process of primary active transport process. Okay, and this one is, you can consider as a secondary active transport. Okay, that means the proton pump, which is embedded on the membrane of the synaptic vesicle, is always open, always active, and gradually when it will active, then it will activate the ATPase, it will show the ATPase activity, and that will make energy from the ATP. That means ATP will turn into ADP, and that will increase the aerobic phosphate, and it will release the energy, and that energy will be used to open the will just pump, will activate the pump, and proton will enter from outside to inside. Okay, and as it is, as the definition of primary active transport, which will be directly attached with the ATP energy. Okay, so this process is basically done by the primary active transport. So initially, the proton concentration was less outside the vesicle, and inside the vesicle proton was more. So again is the concentration gradient and the use of ATP. This is called active transport, and after that gradually it will load the neurotransmitter, and a time will come when, by the influence of electrochemical gradient, from low concentration to high concentration, the neurotransmitter will enter. At that time you can consider as a secondary active transport, because also it is concentration gradient, again is the concentration gradient, and it is not directly connected with the ATP energy, and it is basically influenced by the electrochemical energy. Okay, so this is called secondary active transport process. Okay, so now I will say the primary active transport and secondary active transport, and this is the basic mechanism of the neurotransmitter loading. Now I will say types of neurotransmitter transporter but antiporter, and then you can just find out the particular neurotransmitter and particular neurotransmitter transporter. So if I just if you think this is the for glutamate, for you can say glycine, for you can say a complete neurotransmitter easily. This is the GABA. These are very simple neurotransmitter as well as amino acid. Yeah, so these are the monoamine. So that transporter channel will be for that. That is also another things we can add here plus by adding like the serotonin. You can say histamine, and also you can say dopamine. You can say noradrenaline. Those substances, those neurotransmitter will enter through the antiporter channel, that is neurotransmitter transporter channel. The name will be the muscular monoamine transporter channel. So what would be the transporter channel name for those neurotransmitter? Because these are directly amino acids, and these are basically comes from a single step conversion from the amino acid, and that is serotonin. That is the you can say histamine from histidine, serotonin from tryptophan, dopamine from tyrosine, noradrenaline from tyrosine. So these are also monoamine. So muscular monoamine transporter. Okay, and all the things, all the neurotransmitter will enter through a single neurotransmitter. This is not like that, because if you think this is the dopaminergic neuron, so that would be the BMAT for dopamine. If there is the you can say glutaminergic neuron, so through this channel glutamate will enter. Okay, so now another thing is what will happen actually. Proton will exit and neurotransmitter will enter by the influence of electrochemical gradient. That is the normal procedure. So how many number of proton will come out and neurotransmitter will release? So basically the single neurotransmitter, that is also a amino acid, a single proton will come out and neurotransmitter will enter. Okay, so for this a single proton will enter. Sorry, a single proton will come out. A single proton. And for those and other like acetylcholine and other neurotransmitter, two proton will come out and neurotransmitter will enter there. That means from the inside, two proton will come out or exit and neurotransmitter will enter for serotonin, histidine, dopamine, noradrenaline, adrenaline, acetylcholine. And for glutamate, glycine and GABA, single proton will exit and single glutamate or glycine will enter. Okay, so this is the matter of the neurotransmitter loading process. Okay, so within the terminal, neurotransmitter will produce or synthesized, and that will initially will be available outside of the vesicle. That means initially neurotransmitter is higher in number outside of the vesicle than inside, and a time will come when the BATPase will be activated, and then it will show its ATPase activity, and then it will produce energy from the ATP, and that energy will activate the pump from to enter the proton from outside to inside. That is proton pump, and then it will create a more acidic condition within the synaptic vesicle, or you can say synaptic lumen, and that will create electrochemical gradient. That means charge gradient and chemical gradient, proton gradient. And finally, through this electrochemical gradient, then the transporter, or precisely you can say antiporter channel, will open, and through this proton will exit and neurotransmitter will enter. And by this process, neurotransmitter will be loaded, or you can say packaged, and you can say stored within the synaptic vesicle. One thing is that the neurotransmitter vesicle, when it will be saturated, when it will be perfectly loaded, then it will be ready for docking and release, docking and fusion. Okay, not like that. It will make a equivalent condition from inside and outside, and that will inhibit the loading. That is not matter, because here the secondary active transport, already I said, by this process, from the low concentration of neurotransmitter will just enter into the synaptic vesicle, and time will come, the vesicle will be completely loaded, that is saturated by neurotransmitter, and then it will just come to the active zone, and then it will be ready for the release based on the nerve activation. Okay, action potential. So this is on matter. Finally, I will say what are the factors that will affect, that will regulate, that will control the loading process. So if you just look into the matter very closely, then you will find out the concentration of ATP is a factor. More ATP when will be available within the cytosol, the loading will be more. If you think the number of neurotransmitter will be higher in the cytoplasm, or you can say cytosol, then more loading will be there here. And another things, the proton concentration, that means ion from inside outside, also it will be influenced by some other ion like chloride ion and sodium ion. Okay, so ion concentration is another factor. And you can say also the embedded transporter channel, antiporter channel. The more number of transporter channel, the more neurotransmitter will be loaded. Okay, and another thing, that is your pH condition, that is acidic condition, your ion concentration, your neurotransmitter concentration, your amount of ATP. Okay, and also some vascular membrane protein, that is vascular protein, SB2A and snare protein present on the membrane, and that will also regulate or control the loading of the neurotransmitter. Okay, so these are the factors that can affect the loading process of the neurotransmitter. So this is all about neurotransmitter loading process, or you can say neurotransmitter packaging process, or simply storage process. So thank you very much.